has really changed, or in some cases slightly less time, has really changed the way that these fruits and vegetables look, almost unrecognisable from their wild counterparts. So that's what they look like now. But who's to say that we should stop domesticating now? Surely we need to carry on changing, creating new fruits and vegetables that respond to our needs today. And so this is a pretty picture which shows how some fruits and vegetables might look, you know, with bright purple potatoes. I mean, they're a reality. I'm not sure if they're a commercial success. I'm going to talk about our red-fleshed apple. There's these amazing little stripy onions that have got these candy straps, and that look absolutely fantastic. But at the moment, they're about the size of a thumbnail. And then on the right there's a red kiwi fruit. That was Photoshop. But you know, in those many years of breeding and domestication, we've been concerned about a number of things, but now the challenges of the future are very, very different. We've got to think about sustainability. There's some pictures here just to remind us. You know, the top left on there, that's the phosphorus mines in Morocco, in northern Africa. 70% of the world's phosphorus is in one small place in Africa. New Zealand and probably Australia will have to import nearly all of that. That is not sustainable. So how do we deal with that? Do we make plants which need fewer nitrogens in order to grow? Water, 70% of the water the fresh water that we use is used to irrigate horticulture. And also, you know, we're going to hear next about mechanization. Here's a little hamster ball that some Spanish have developed to go up and down the rows and try to help horticultural practices. You know, how can we help domesticate plants so that they can enable some of these mechanizations? And light, these are some plants that are under light. A lot of plants need light, and light levels change throughout the year, and part of the domestication is disassociating the need of plants to need different levels of light during the summer. Can we enable things to grow at different times of the year? And so these are all challenges that we haven't necessarily worried about in the past, but in the future we need to think about them. So can these be components of our domestication project? And there's also a fascinating discussion to be had about commodity versus premium. You know, we're asking people to invest research dollars into some of these breeding programs. And so you've got to say, well, where's the return on the investment? There's no use investing in trying to increase productivity in something which has got a very small yield, because that's not really going to have a value proposition. If you're working on a big row crop, wheat, maize, rice, whatever, then small increases in productivity have huge returns, and so that makes sense in a commodity. But if you're working on premium, then the targets that you need to chase in order to justify the research dollars are slightly different. And certainly in New Zealand, that's where we put a lot of our effort, certainly in the fruit world. And I'm going to be touching on some of the research that underpins that for the rest of the talk. So this is the big sort of dilemma that we have, or opportunity maybe, in that we want to invest research dollars, but we want to try and make sure that it impacts on the industries that we work for. And so in science, I learned a few years ago there are only two questions, so what and who cares? And for me, this little three-dimensional graph answers a lot of those questions, because what I'm going to talk about for the first half, or the remainder of the first half, is about some of the traits, some of the biology, some of the things that we think are going to enhance the premium that growers can get in the crops that we work on. So that's the science, that's the technology. But then, you know, so what? You know, we've found that fascinating information out. What are we going to do with that information? If we just publish it and then move on to the next question, then nobody's benefited. So for me, using emerging and existing technologies to work out how we can take that knowledge and integrate it into a breeding program is about the so what. How can we take that knowledge and get it actually implemented into a breeding program? And finally, who cares? Well, hopefully we've had a chat to the breeders and the growers before we embarked on some of this research. And the things that we're working on are things that are really important to them, the things that are going to make a step change in the industry. And so hopefully that's guided our decisions on the trait-focused work in the first place. So for me, traits are about value, and I think it's important to think about production traits, those that affect the growers, but also consumer traits, those that affect the output traits, because if you're growing fresh fruits and vegetables that aren't processed, you can't do much to it. You've got to grow it and then you've got to sell it, and so you really want to try to use technology to make sure that that particular product that you've got has all sorts of attributes that are going to help the value of it. Its appearance, the sugar acid balance, the flavours, the storage and shelf life, the convenience, the health. These, we're told from our megatrend analysis, are the things that drive consumers and their willingness to pay a premium. Whereas if you're growing them really things that are important to you, apart from people wanting to sell your stuff, it's about disease resistance, it's about productivity. In our tree crops, rootstocks have a huge effect on the way that we manage our crops. And I've touched earlier on, thinking more and more about some of these sustainability drivers, water use and nutrient efficiencies. So these are the sorts of broad areas that we think about. This is a quick example of the rootstock I touched on. Apples are really, really lucky. Many years ago in France, they found this dwarfing rootstock. It's a rootstock that significantly affects the shape and architecture of the scion. These are two Braeburn apples, genetically identical scions, but they're on different rootstocks, and you can see the difference in the architecture of those plants just by grafting them onto a rootstock. And that affects precocity, so you get a crop earlier, affects the stature, so you don't need to require the same amount of mechanization in order to harvest the crop. So lots and lots of benefits. They've got fewer leaves, so they require less water. I mean, the advantages of these rootstocks go on and on. You can have disease resistance in there. And we're actually using genetics to understand the changes in the DNA that have taken place in those M9 rootstocks so that we can understand why they have this altered architecture. And we're going to use that information to help develop apple rootstocks, but we can also use it in other crops as well. And I'm not going to go in any more detail about that, but I will talk in a lot more detail about that, the red apples. So here we have some pictures of naturally occurring red-fleshed apples that were found in Kazakhstan. There's so many things that are wrong with them. They don't store, they've got terrible texture, their flavour's not very nice, but they look pretty amazing. And alongside disease resistance, these red-fleshed apples were the #1 target of our apple breeding program. And so we were keen to see whether we could do some research to help support the breeding program. And red's pretty fascinating stuff. This is some work from Kathy Martin at the John Innes Centre in Norwich, a colleague of mine, who showed in work with tomatoes that if you increase the levels of anthocyanin, you can actually reduce the incidence of chronic disease. So this is a tumorigenic mouse. So basically, these are two lines here. One of them is a number of mice that are fed on a regular diet or on a diet supplemented with tomatoes, and you can see that the two lines are the same. But this purple line here is what happens when these mice that are susceptible oh, that's going to be good. And too much coffee, I think. We're fed on these high anthocyanin tomatoes, and you can see that every time there's basically this is what a little mouse's diet, so you can see that fed on these high anthocyanin diets, they lived a little bit longer. Nobody quite knows why that happened, but certainly a fascinating observation. So as well as looking pretty gorgeous, it may be that some of these anthocyanin compounds have got health benefits as well. And so we embarked on a number of things. First of all, we found the gene that we believe was responsible for the red-fleshed apple, and then we were able to locate the position of this gene. So I'm going to talk about genomics, and so this here is a chromosome. And a chromosome has basically got lots and lots of genes on it. Apple has got about 17 chromosomes. Each would have about 5,000 to 10,000 genes. And so here is the gene here that's responsible for the red-fleshed apple. But we looked a little bit more deeply, and we were able to not just find the gene, but look at the change in the DNA that was responsible for making those red-fleshed apples. Because in conversations, sometimes people say, tell me about the gene for red flesh. And actually, all of the apples, even the ones that have got white flesh, have the gene. It's there in the chromosome in the same place. The only difference is whether that gene is turned on or not in the red-fleshed apple. That's what's changed. So there's no new genes, there's no new genetic information. What's changed is when the gene's turned on or not. And basically, genes, you may have seen them before, they look like this. They're little pointy things, they've got a beginning and then little arrows. And what happened is this single apple in Kazakhstan was a small piece of DNA. This is a piece of DNA here. I haven't written it out, but it would have been about 20 DNA molecules, amino acid nucleotides long. And in the white-fleshed apple, there's just one copy of it. And in the red-fleshed apple, somehow it's gone from being one copy to being 6. And when DNA is copied, often the machinery that replicates DNA makes mistakes. And here it made a little mistake, and it did something 6 times instead of once. And that led to the red-fleshed apple, and the change was as simple as that. We've gone on to show what the mechanism is. And that's been quite useful, actually, because at the time the breeders were interested to see whether they could separate the red fleshed apples from the red leaves, because what they knew were these red apples also had red leaves. And so they were breeding and selecting and so on. And when we knew the mechanism that this red fleshed apple, the change in the DNA, we were able to say, look guys, the way it works, it's always going to be red everywhere. It's just what it does, and it will be almost impossible to separate those two. So if you're really interested in having something that's got red or pink flesh but with green leaves, then you're going to have to find another allele, another version, another chain, another source of that variability, because the one you're working on, the one from Kazakhstan, is not going to do it. So that information is helping our breeding program, and I'm going to come on and touch on the breeding in a few more minutes, but I want to just talk a little bit about how we've used genetic modification in this research. And we've used GE not to make a product, and it's really important that what we've done is take that gene and we've put it into Royal Gala, and we made a genetically modified Royal Gala, and we've used that for research. And it's told us things that are really helping our breeding program. But the way we've done it isn't to produce a product. We would never sell this. We wouldn't be able to. We wouldn't get a license, because the technologies that we've used, we wouldn't be able to license. But we've got a research license for them, and they can tell us things. So here are the genetically modified apples in tissue culture. They grow, and often apples, when they're modified, we take a callus and we modify it, and it produces shoots, but getting it to root is a little tricky. So what we do is micrografting. We take the little shoots and we graft them onto M9 rootstocks. We then grow them in growth rooms, which are hot. We raise them up to 30 degrees. We give them lots of carbon dioxide and lots of light to try to get them to grow really, really quickly. We have to spray them actually with paclobutazole to reduce the internode length, otherwise they grow out of the root. And finally, after about a year and a half, 2 years, we actually got a crop of genetically modified apples. So this is, on the right-hand side, a Royal Gala. On the left-hand side, Royal Gala with one extra gene. The changes in the DNA are less than 0.01%. It's just a tiny addition of an apple gene, actually, that's been overexpressed, and it produces these stunning apples. So we were able to do some experiments with those genetically modified apples, because they were almost identical to Royal Gala. Except they didn't look identical, but genetically they were identical, and so we could do all sorts of experiments on storage and flavour, and we could do some feeding experiments as well, and say, how do those red Royal Galas differ from the regular Royal Galas? Because all of the changes that we see are due to that single gene and those elevated anthocyanins. And when we do eventually breed an apple that's red, then we're going to have to know how to store it. We're going to want to know whether consumers like it, whether it's bitter, and we can use this genetically modified apple to test some of those things, because if we wait for the breeding program, which is a few more years away, then once the cultivar's bred, then if we start doing these experiments, there'll be another 5-10 year lag before we can start really ramping up commercialization. Whereas using this genetically modified apple, we found out some really interesting things. This one is probably one of the most shocking things. You can see here the height, the width and the weight of the fruit is largely unchanged. Great news, we're not going to affect yield or productivity with these high anthocyanin apples. The firmness is pretty much the same. But look at the storage. You can see here, when we take these red apples, these genetically modified Royal Gala apples, and store them under conventional temperatures and atmospheres, they all go brown. Whereas the control here, you can see it's missing, because Royal Gala has got a post-harvest storage regime which has been optimized so they don't go brown. So this is really useful information because we need to now change the way that we store these red apples, because if we use the protocols that you use for Royal Gala, they're going to go brown. Now, I don't believe that these apples will go brown. We'll just change the way that we store them. We may need to store them at a slightly warmer temperature, or we maybe need to change the atmosphere. But isn't it good that we know that now, not once we've bred the new variety in 10 years' time? So really useful information. So now we're developing the post-harvest methods that are needed for a variety that hasn't even been developed yet. But we believe that these things, the information that we'll get, will be translatable because the genes that we're using for GE are the same genes that the breeders are selecting for in their breeding program. We also have eaten these apples. We weren't really allowed to eat them in New Zealand. We put in several applications, but it was awkward and it was an election year. We all know how volatile these things can be, and so we were told to keep our heads down. And so we're in a dilemma. We've got a pile of genetically modified bright red apples that we're desperate to eat, and we're told not to do anything stupid. So what we did was buy some tickets to Sacramento, and we flew over to the States, and we got 50 Americans to eat the apples. And we actually employed a company to ring up people in California. They're very liberal over there. And we said, look, can you find us some people? So they rang round, and there was a professional company that did this, and they said, first of all, we want regular apple eaters. That was proved a problem. We had to have somebody who'd had an apple in the last week, so that eliminated about 90% of the Sacramentans. The remaining 10% really were very happy to eat our genetically modified apples. We didn't tell them they were red. They came along and they sat in these chairs. They signed a bit of paper, which was very kind of them, said they wouldn't sue us if anything untoward happened. But we said we wouldn't show our faces. And we gave them these black glasses so they couldn't see what they were looking at. And we gave them 2 bags. And in the bags was either a genetically modified Royal Gala or a Royal Gala that was unmarked. And they reached in blindly and tasted one and the other. And then we asked them whether they could taste the difference, because those high anthocyanins are phenolics, they're phenylpropanoids, and tannins are also phenylpropanoids. And they're the tannins are the compounds that are responsible for that bitterness or astringency that you have if you have a cold black cup of tea. And in some instances that can be unpleasant. And so we were really keen to know whether these apples were going to have any negative taste attributes. And so we fed these apples to the Sacramentans. We did some naughty things as well. We gave them 2 identical slices from an apple and asked whether they could spot the difference, and half of them, of course, could. And they much preferred the first one to the second. Reassuringly, when we gave them 2 different varieties, they could spot the difference. And so, working with consumers is a challenge, and we're really fortunate in that we do have a whole team of people who are consumer experts. And so, anyway, the bottom line was that they couldn't spot the difference. The only thing they could tell was that there was a slight difference in texture, and that was quite reassuring because we knew that the red apples were slightly softer. And the reason we think that is because they're red, they're bright red, and they get much hotter. It's just physics. Dark things get hot. And so they obviously, during their development, are slightly warmer, and so we think that they just develop slightly quicker. We've got experiments on the way to test that. There is a debate. Auxin, one of the hormones that plants has, moves around the plant, and anthocyanins also affect the ability of that to move, so it may well be that the anthocyanins affecting auxin transport. So that's the debate we're having, my colleague and I. But we do know that there was a slight difference in texture, and reassuringly the consumers spotted that. But no bitterness, no astringency, no tartness, and so that was quite exciting. And when we got them to take their goggles off and look at the apples, they all bounced around really excited. Oh, I'm going to do this, I'm going to do that, I'm going to put it in a salad, I'm going to put it in a pie. And it was really interesting to see the consumer's response to that. And the consumer scientist that was with us said she'd never done a taste trial with people where they were all so positive. She'd never been to America either, so I don't know which it was. The other thing we've done is start to look at the role that these anthocyanins might have in diet. This is following on from work that Kathy Martin's done with her mice. The first thing we did was look to see if there were any changes in the expressions of genes associated with inflammation. And so a lot of the research is suggesting these phenolics, these antioxidants, may actually act to enhance the immune response in ways that I really don't fully understand. Anyway, this is some expression analysis that shows that some of the enzymes in your stomach, in the duodenum actually of mice, are changed slightly when they're fed on these high anthocyanin apples. But for me, this isn't quite as interesting as the fecal output. This is a fascinating experiment. It just so happens that when we work on mice, we use what are called calorific cages, and basically they measure everything that goes in and everything that comes out. And so we really still haven't got to the bottom of this data, but what we have done is repeat this experiment twice, which is a good thing to do. And basically what we've observed is that if you fed either a standard diet that we feed mice, or one supplemented with Royal Gala, then the flow of food through the mouse remains largely unchanged. However, when these mice are fed on this high anthocyanin diet, then it seems to go through a little bit more quickly. So we see an increase in fecal output. And actually transit time, which is really what we're measuring here, we believe, and also the composition of bacteria that exist in the stomach are really important for our nutrition. And more we're beginning to understand that actually it's the bacteria in your stomach that contribute to nutrition more than the nutrients themselves. And so we're really still early days here, but beginning to understand maybe how some of the anthocyanins in our diet are affecting our gut microflora, and then in turn having some of these positive health benefits that our colleagues have seen. So that's all I'm going to talk about. How am I doing for time, by the way?
About 5 minutes.
5 minutes. Right, I might...
If you're very good.
What I'm going to do, I'm going to skip, I think, vitamin. I was going to talk about, I think you said apples and orange kiwi fruit. I thought I'd talk about vitamin C in kiwi fruit, because vitamin C is a fascinating thing, actually. People need it, and we all know, because we've all emigrated at one time or another, or have relatives who have. And so it works as an antioxidant, but also as a cofactor, and so that's why in collagen biosynthesis, if you don't have ascorbate, then you have problems with. This is one of the nicer pictures when I Google imaged scurvy. Most of them were pretty horrible. Anyway, vitamin C is an important part of our diet if we want to understand scurvy, but also moderate levels of vitamin C or ascorbate also help cognitive behaviour, and a lot of people, and there is some evidence to suggest that if you're not feeling well, or if you're going to get the flu, then if you elevate the levels of vitamin C, it has some positive effects. And so, lots and lots of examples where vitamin C's got quite an important part of our diet. And so there is also a fascinating debate going on, trying to decide whether it's better to have a pill or to have something in the whole food matrix. And there is also anecdotal evidence. These experiments are pretty hard to do, but they do suggest that if you have an efficacious compound like ascorbate, but it's in a food, then it's better than having it in a pill. For me, I'm sort of a gut feel person. It just feels if you can get the same thing from a food, then that would be great. And so at the moment, my job is about kiwi fruit. I'm helping breed kiwi fruit from the Genomics through the new cultivars. And so I thought I should at least talk a little bit about kiwi, who are fascinating because the levels of ascorbate are incredibly high. I mean, in fact, in Asia, if people feel the need, or if you were to ask them, where do you get your vitamin C for, they would say they'd get it from kiwi fruit. And that's partly a marketing exercise, but it's partly because the levels of ascorbate and vitamin C in kiwi fruit are really high. They're twice that of an orange. And if I just show you this is the cultivar. This is the green kiwi fruit. This is the one that's considered high in vitamin C. So that on this graph, it's tiny compared to some of the germplasm that we've got. That one on the left there, Actinidia eriantha, has got almost 1,000 mg per 100 grams of kiwi fruit. So that's really high levels of ascorbate. And so not an edible kiwi fruit, but in our breeding program, we could potentially have a super high vitamin C kiwi fruit. And kiwi fruit are the perfect thing to work on vitamin C because they're high in the stuff. And so for a few years, we've found the various enzymes that are responsible for making vitamin C. And it's actually fascinating. Who would have thought, we've been working on, we've known about vitamin C for a long time, but it was only in 2007 that my colleague, William Lang, found the final step responsible for vitamin C biosynthesis. So that's taken a long time. And these are the various enzymes. I'm going to skip through this quite quickly, that are responsible for making vitamin C. But what we've been able to do is identify this particular gene here, this phosphatase, which is actually the rate limiting step in the pathway. So if you want to increase vitamin C, all you need to do is regulate or change the expression of that gene, and you'll get more ascorbate. And we've delved down a little bit more, and in the same way that we found out how red apples make themselves red, we've now found out how high ascorbate plants make lots and lots of vitamin C. And it turns out to be a really fascinating story. I can't go into the details, but we've all got genes, about 35,000 of them. Apples have got about 35,000 genes, and they all always start with a methionine. It's an amino acid that begins a protein. But in vitamin C, for some reason, it starts with a threonine, a different amino acid. And we think it may have even a different translation initiation system. And this is fascinating science, but we think it's because ascorbate's really, really important. It's important for plants. In fact, if they don't have it, they die. And we think that what plants have developed is a special mechanism that in times of stress, when you turn off lots and lots of the things that you normally do, you keep a few key things going, and one of them is keeping ascorbate going. And so by understanding how the plants are making this, we now think we can work out how we can start to help domesticate and manipulate this. And so we've got a few experiments here where we can see this is normally the levels of expression you'd see in ascorbate. But if we make single DNA changes, a single nucleotide, we can change that and we can elevate the levels of ascorbate in test systems. So really, by understanding how these compounds are made and how they're regulated, it's giving us a deep insight into the mechanisms that control them, and that knowledge can then be used to help breed new cultivars. And I'm going to talk about the mechanisms in the last few minutes, if I've got them. Yeah, cool. So these are kiwi Victorian, and I'll skip across that. So one fascinating thing about plants is, and us actually, where the DNA is much more similar than you might imagine. This is, the rest of the world calls this a stadium plot, because it looks like a stadium. In New Zealand they call it a rugby ball plot. And basically each of the rings, each of the colours is a different genotype. And so on the outside is kiwi fruit, the genome we just sequenced recently. And as you come in, there's tomatoes and potatoes and grapes and cucumbers. And so, as you can see, we come right down into the inner circle, which is a little weed that grows in the ground called Arabidopsis. So all these plants are really very, very different. But here we're showing the number of genes that are similar between all of these plants. And here, we're just showing the number of genes that are different. And so the message is that actually, although the plants might look very, very different, the DNA inside them is incredibly similar. And that means that we can find genes in crops really, really easily. When we sequenced the apple genome, for instance, we found that the genome had been duplicated 50 million years ago. It had got this odd, so instead of having one genome, there was two. And because the DNA looks very, very similar, we were able not only to spot where our beautiful red-fleshed apple gene was, but on a duplicated chromosome, there was another gene responsible for making these beautiful pink apples. And we found that by sequencing the genome. And now we can use that in a breeding program to select these beautiful pink-fleshed apples alongside the red ones. So, the technologies that we use. Once we've got this knowledge, we can use conventional breeding. We can follow the inheritance of single markers and try to predict what plants are going to look like. But we can also use transgenics. You know, this is scary stuff, but really what we're doing is taking DNA, sometimes from an apple and putting it back into an apple, but the mechanisms that we do insert this piece of DNA randomly, and so people have concerns about whether these random integrations have an effect. But there are emerging technologies that allow you to be subtle still, and when we talk about keyhole breeding, about the idea that we can go in and really make quite simple changes. Zinc finger nucleases and talons, these are technologies, but really what we're able to do now is use these techniques to just modify a single nucleotide, not a whole, and it's targeted as well. We can work out the precise positions in the genome where we might insert or remove these genes, and so the knowledge that we've got starts to become more and more applicable with these techniques. But you don't need to use GE. There are other things. They're a bit harder and a bit slower. But mutagenesis, you can expose crops to radiation. Canola, for instance. Canolas, Canadian oil, low acid. It was mutagenized. It was high in an acid which made it impalatable. They mutagenized the crop to remove the nasty acid, and now we've got canola grown everywhere. These beautiful oranges, these Valencian blood oranges, haven't been bred. They weren't crossed. They were just selected as natural sports that existed from oranges that are hundreds of years old. And the reason they're red is because a transposon, a piece of mobile genetic information that jump around the genome all the time, this particular Valencian blood orange, a transposon, inserted itself near that gene, the same gene actually that regulates apples to become red. Well, that was turned on, not using GE, but using a transposon. So how might we harness the ability of those mobile genetic elements? And this is some fascinating stuff. We talk about the DNA, but what about, DNA doesn't sit there by itself. It's coated in all sorts of chemicals and proteins that protect it. And those themselves can change. And so here we have a bunch of grapes that was grown in Marlborough. Sauvignon Blanc, they're supposed to be white, but occasionally, very occasionally, you see these little purple bunches of grapes popping up, and you can actually propagate those and grow them. And it's not changes in the DNA, we believe, but changes in the epigenome, the stuff that's around the DNA that's changing it. And they can lead to quite fascinating phenotypes. And so, as we learn to understand the epigenome, how might we manipulate it so we can create novel varieties and cultivars that have got these additional benefits that we're interested in? And then, of course, there's breeding. I've got a couple of slides. Breeding is a fascinating pursuit, really, and I think that genomics research can contribute to that as well. I mean, hypothetically, if this Dutch guy here was breeding tulips looking for a red one, he doesn't need any fancy pants genomics to spot the red tulip that's growing in the middle of the field. But just imagine if he'd had to wait 6 or 8 years for that tulip to flower. Then maybe it would have been useful if you could have predicted in year 1 rather than having to wait 4 years. In kiwi fruit and apples, we have to wait even longer for them to flower. But what happens if the traits are not that easy to see? What happens if it's a flavour? Or what happens if it's storage? Then actually the cost of phenotyping that trait becomes really, really expensive and really, really slow. And so after a time it becomes feasible and actually financially attractive to actually look at the DNA and actually predict whether it is going to have a phenotype or not. And what happens if it's not one gene, but several genes that are involved? Here, on the right-hand side here, this is a chip, a SNP chip. Each one of those dots is a different gene from apple. And the colours really indicate whether that particular individual has inherited a gene from the mother or the father, and you can see there's all sorts of different colours, and that's because when you cross an apple, as everybody knows, that the DNA gets mixed up and the progeny never look like the parents. And so we're able to look at these slides, and we're able to use statistics. We use actually the statistics that breeders use to actually work out breeding values and to make a statistical judgment on the ability of that particular seedling to store, or whether it can taste good, or whether it's going to be crispy. Complex traits that are multigenic, but we're able, using new technologies, to actually predict these things using statistics. And the thing that's really made this possible has been a step change in the way that we look at DNA. This is a timeline of how DNA is sequenced, which is our way of visualizing DNA. And when I was doing my PhD down here, we used manual slab gels, and we looked at a few hundred bases a day, and it took forever, and it cost a fortune. Now, with these massively parallel techniques, it becomes so cheap. We've had to put the data on a logarithmic scale to really emphasize how cheap it has become to sequence people's genomes. The first person probably cost $3 billion to sequence. We could sequence somebody's genome today for about $2,000–3,000, and that will only get cheaper and cheaper. And so, really, we're at a point where we need to decide on the traits that we want to breed for, and then we need to use the technologies that we can to really translate that information into a breeding program. And I show a few kiwi fruit here because we're really interested in a red kiwi fruit. We'd love one. But terrible things happened in New Zealand around the world. Pseudomonas syringae actinidia, PSA, has come along and it's killed a lot of varieties. It's killed this one here. This was the one that we sequenced the genome of. It's gone. And so as nearly all of our red breeding program, they were all susceptible to PSA. But within our germplasm, within our collections of wild stuff, we've got these smooth-skinned agouti kiwi fruit that seem completely resistant to kiwi fruit. And so how do we start to take these wild species and cross them into some of the cultivars and get varieties that are resistant to PSA and maybe have all these desirable traits that we know consumers will pay a premium from. And so really we're throwing as much information as we can to understand the traits, but also using all of the technologies, be they GE or non-GE, to accelerate our ability to integrate that information into our breeding programs. And there I will stop.
Well, Roger, what a fascinating presentation.